Okay, everyone, let's take a look at linkage. I've been talking about linkage, alluding to it for 5 lectures now, I think. And now we get to start talking about it. So in this short video, I am going to just introduce linkage and show how it leads to a different genotypic ratio in the gametes than what we were used to seeing with independent assortment, right? When all the gamete genotypes were formed in equal ratios. Well, linkage is going to disrupt that, and it's going to cause a change in genotypic and phenotypic ratios. So we would end up with modified Mendelian ratios, or ratios that are non-standard. So I'm going to go over that, and I'm also going to go over an important way to symbolize linked alleles that'll be necessary moving forward in the course. It will be necessary for you to understand moving forward in the course, especially during the next 2 lectures. So to begin our discussion on linkage, let's look at 2 genes in Drosophila. We're going to look at the heavy gene and the brown gene, which we've already looked at in one of our examples on gene interaction. So the heavy gene will symbolize with an HV. We'll use a plus for the wild type allele. And homozygous flies here, recessive flies here, will have the heavy phenotype, and it refers to the veins of a Drosophila wing. I don't work with Drosophila, but let's say this is a wing and it has these little veins here. So what the heavy means is, instead of having thin veins like that, the veins are really, really thick. Okay, so normally the veins are thin. Okay, so heavy refers to the wing veins of the fly. Now brown, we've seen that already, but just in case, let me put the genotypes down here. So we have red eyes. Again, I'm just saying red, not brick red, but red and brick red are synonymous for talking about Drosophila eye color. And the homozygous recessive condition leads to brown eyes. Now, okay, so these are linked, and then we're not talking about gene interaction here. These control different traits. So I just don't want you to get confused and think we're going back and talking about interaction between brown and heavy. No, these are different traits, but it's a good example for talking about linked genes that are close together on the same chromosome. And to get set up, let's just imagine a cross between these flies here. So wild type for heavy, mutant for brown. So we could call these flies thin and brown, and over here we could call these heavy and red. And sometimes, it's very often just to say the mutant phenotype. So you could call this brown flies crossed to heavy flies. I will try not to do that. I will try to list both the wild type phenotype and the mutant phenotype in each fly, each parent of the cross. So if we did this cross, well, we already know what the F1 genotype is going to be. And I don't want you to get thrown by all these complex symbols here. This is no different than what we have done in the past. I can symbolize, let's say, use A for heavy, and then use B for brown. And you know, if you cross something that is homozygous for big A and little b with a strain that's homozygous for little a and big B, you know that all the offspring are going to be heterozygous for both pairs of alleles. So we know that all the offspring are going to be heterozygous for both pairs of alleles. Okay, and because they have a wild type copy of each of an allele for each of these genes, the phenotype is going to be wild type, which we can specify as thin and red. So thin red flies. Now, when we cross these F1s to another F1, we don't have to worry about X linkage here. These genes are not on the X chromosome. And again, we don't need to worry about incomplete dominance or codominance. Heavy is completely dominant to the recessive allele, and the wild type brown allele is completely dominant to the recessive brown allele. Now, so if we cross this, we would normally predict a 9:3 to 3:1 ratio. So 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16, where these guys are both dominant for both traits. And this is dominant for one trait, and these guys are dominant for the other trait. And these are both recessive. However, because these are linked, and because the gametes aren't produced in 4 different types, which are in equal ratios, we don't end up with a 9:3:1 ratio like Mendel saw in his dihybrid crosses. This being the F2 generation down here. We end up with a different ratio. And the ratio changes depending on how closely linked these genes are. So let me just put down a brief diagram of what these could look like on the chromosome, say in the parents in the P1 generation. So here are the genotypes we started with. Now, if we just take a nucleus from a somatic cell, don't even worry about meiosis and tetrads and things like that. And we'll assume the chromosome is condensed. So I'm going to put the BW allele right here and the HV plus allele right here. So I'm showing these being very, very close together. So Drosophila is diploid, and we know, okay, there's a homologous chromosome that matches this one. So I'll diagram that somewhere else in the cell. And we know that, okay, the brown gene will be here, the heavy gene is right here. So they're going to be at the same positions on the homologous chromosomes. We know from the genotype that this allele must be BW, and this one is HV plus. We could do the same thing for that other parent. So, but here, the alleles are BW plus and HV. Okay, now, so this is important because I can now introduce a new way to symbolize genotypes. Now, in this new way, this method is going to allow us to see how the alleles are arranged relative to one another. So we draw a line, and all the alleles on one chromosome are put above the line. So HV plus is with what? BW or BW plus. And this fly is with BW. So we can see that right here, HV plus BW. Now, we put the genotype of the homologous chromosome below the line. HV plus BW. And it doesn't matter which chromosome you choose to put on the top or the bottom. It doesn't matter. You just need to put one up above the line and one genotype below the line. And here they're identical, but it doesn't need to be the case. These won't always be identical, which is the value of showing the arrangement like this. Okay, so we can do the same thing for this parent. HV is with BW plus, and HV is with BW plus. Okay, so when these flies mate, each offspring can inherit one of these chromosomes. This one, or this one, or this one, or this one. And even if, we're going to talk about recombination later, but even if recombination should occur between these two genes, it would be so, I mean, it wouldn't matter because the alleles are identical. So the offspring are going to inherit an HV plus and BW. And over here, the other chromosome they're going to get is going to have HV with BW plus. So now this is important, right? Because this notation system here with the line allows us to immediately look at this fly and say, okay, so the wild type heavy allele is with the mutant brown allele, and the mutant heavy allele is with the wild type brown allele on the other chromosome. So one chromosome, wild type heavy with mutant brown, on the other chromosome, mutant heavy with wild type brown. And when this goes through meiosis, I can diagram this quickly. Let's assume meiosis is occurring in a fly with this genotype. You could pause the video right now, and what you could do is diagram what do you think a tetrad would look like in this fly, and you could label the positions of the genes and put the alleles down, and I'll show you what that looks like here. So let's assume this is metaphase of meiosis 1. Now here we have on this side, I'll put this guy, this chromosome, HV and BW plus, and I won't label this one, this sister chromatid, it just will get too messy. And over here I'll put HV plus and BW. Okay, now in almost all of these, HV plus, I mean, after metaphase, after these guys segregate from each other and go through both rounds of meiosis, 99% more or less are going to have a genotype, or they're going to get a chromosome which has HV next to BW plus, or you can see up here HV plus next to BW. Okay, so almost all of the gametes, this fly with this genotype, almost all of the gametes will have this combination of alleles, HV with the wild type brown, or this combination of alleles, wild type heavy with the mutant brown. So if you notice, we're missing something, right? When we were talking about genes that are sorted independently, well, we would have predicted 2 more gamete genotypes. So let's assume 99% are going to be this type. So if these genes were sorting independently, we would have expected some of the gametes to have a genotype where we have HV plus and BW plus, so both wild type alleles next to each other, and some where we have HV and BW next to each other, both mutant alleles. We would expect all combination, genotypically, all genotypically possible gametes, gametes of all 4 genotypically distinct gametes produced in equal ratios if these genes were sorting independently. Because they're linked, well, most of the gametes are the genotype that, as it occurs on the chromosomes in the parent, right? So in this case, 99% are these 2 genotypes, and most of them, I'm just guessing right here, we're going to figure out a way to actually calculate this number very soon. In a very small percentage, let's say 1%, are going to be this combination. So where's this 1% coming from? kind of jumped a step right here. I started by saying almost all of them are going to be here, this type. And I said, well, okay, maybe not all of them. So what happens is during meiosis is sometimes we get recombination. So an exchange of genetic information between these paired homologous chromosomes. And another word for that is crossing over. And depending on where the crossover occurs, we could end up with a case where the HV plus comes over here and HV goes over there. And if that happens, we will end up with a small number of gametes with these genotypes. And this does happen. And the level at which crossing over will happen between two linked genes is dependent on how close the genes are to one another on the chromosome. If they're very close together, then recombination doesn't happen that often, and you get a very small number of these recombinant gametes, we'll call these. If they are very far apart, well, then recombination or crossing over is much more frequent between them, and you end up with a larger number of what we'll call recombinant gametes. But we're going to get to recombination next week, so in upcoming lectures. So we'll have at least 2 lectures talking about recombination and determining distances between genes and mapping the position of genes on chromosomes. Okay, so I hope you're finding these videos useful. Again, this is my first shot at doing videos with this format. I do need an updated phone, but I hope they are proving useful for at least some of the students in the class. Okay, so see you next week or office hours, or if you have any questions, email me, or other than that, see you next week.